I'm Kshitij. I'm a graduate student here, and as everyone sort of settles into their food coma, I'm going to be talking about some of the work we've been doing around using machine learning for predictive gene circuit design. So we're a synthetic biology lab, and we primarily focus on building these DNA-encoded programs. And once my clicker works, okay, I think I got it. Yeah, I think it's fine. Yep. Okay, great. Yeah, sorry. So we think about building these DNA-encoded programs. And now DNA-encoded programs have a lot of applications across the healthcare space, from the bioproduction of biologics like antibodies to delivering gene therapies, or even producing cell therapies where you take immune cells and engineer them to give them new functionalities to fight diseases. And as synthetic biologists, we think a lot about what goes in here in the center of this orange circle, the genetic programs that you can put into cells to give them the ability to sense external molecules, such as disease cells, and then once they sense those, integrate those signals and then produce some kind of a therapeutic response, such as a signal that basically kills the disease cell or the cancer cell. And a very common, very popular example of this is the constitutive expression of a chimeric antigen receptor, or CAR T cells, which do exactly this. They sense target cells and then produce some sort of signaling response that kills the cells. And CAR Ts have been very successful for some kinds of cancers, but they fail in other kinds of cancers due to a number of failure modes. But a couple of popular ones are this overactive signaling response, where the signaling just goes crazy and the immune system goes out of whack and starts attacking the body's own cells. Or simply, the CAR just doesn't work very well, and it binds to these off targets and still produces the response that kills healthy cells in the body. So in thinking about genetic programs that could fix this kind of thing, you can think of a really simple system that essentially uses the same tumor ligand as an input, and only then turns on the expression of your CAR gene that then has to go and bind to a second signal also expressed on the target cell. And this kind of combinatorial encoding gives you some sort of specificity. And while this genetic program looks simple, it requires the careful balancing of a lot of different parts. So you need to tune the expression level of these components in your program. You need to tune the strength of the activation. Too high of an activation strength, and you might end up with overactive signaling. And a bunch of other parameters that you need to change. In fact, even if you consider a simpler circuit or a simpler genetic program, something that binds a drug and produces an inducible response and turns on the expression of, let's say, GFP. So the circuit on the right is how you would write a genetic program to sort of achieve that functionality. And we'll talk about that more in a second. But if you consider all of the things that you could change, there's like 10 independent features here that control the behavior of this circuit. And if you pick just 3 or 4 parts across each of these categories, you'll end up with 166,000 combinations that you need to test. Now, unfortunately, our ability to test these circuits has been really limited. So you build these circuits one at a time, but then you don't get the quantitative understanding that you want to really learn how to build these circuits. And so we hypothesized that if we had the ability to generate a large library or a bunch of these combinations all at once and measure their responses directly in the same experiment, then this would be a great problem for AI to solve. So you could generate a lot of data and then use AI models to predict how a gene circuit is going to behave just from its constituent parts. And so in order to do that, we developed this platform, and we like to call it Classic, which uses a simple assembly scheme where you sort of give it the input parts along with a library of small sequences that we call barcodes. And it assembles all of their possible combinations in a single assembly step. And during this assembly step, the cells will take in these genetic barcodes at random. And following that, you do step 2, which is long-read sequencing using Oxford Nanopore sequencing to figure out what constructs are linked to what genetic barcodes. And you create this sort of construct to barcode lookup table, which is great because these barcodes are really useful once you deliver your programs into cells and then sort them in some way. So for instance, if you're producing a fluorescent protein response, you could sort them based on their expression using a cell sorter, and then use next-generation sequencing on just the barcodes to figure out all the variants that are present at a particular expression level. And so you get the behavior response of the barcodes, and you know what barcodes are linked to what circuits, so you can combine these two modalities to sort of learn what circuits are behaving in what way. So we decided to test this out with the simple inducible program that I talked about a little bit earlier. So this is a 2-gene circuit that's really popular. And in essence, it's very simple. The first gene produces a transcription factor. Now, the transcription factor goes and stays in the cytoplasm until you add a drug or a small molecule, which then binds to this protein and sends it into the nucleus. In the nucleus, it can find its cognate DNA binding sites and turn on the expression of a reporter gene, which in our case was GFP. So for engineering this, what we're really interested in is measuring 2 responses for every circuit: its expression level in the absence of the inducer, which is its basal expression, and its expression in the presence of inducer, which is its induced expression. And we're particularly interested in engineering circuits that have a high fold change, so they have a high induced response relative to their basal response. As I mentioned before, we changed a bunch of features. Doesn't matter what for now. I'm happy to talk about this in more detail if someone has a question. But we produced 166,000 different circuit combinations using our approach. We put them into cells and put it on a flow cytometer, which is this set of histograms in the middle here. The dotted line is the behavior of the library without any inducer, and the solid green is the behavior when you add the inducer. So we sorted the library twice, once with and once without inducer, and got 2 measurements for each of our 166,000 circuits. And at the same time, we built 40 of these circuits from this library out at random and measured them directly on the flow cytometer so that we could compare the fold changes of the actual variant's behavior, which is on the x-axis here, and the fold changes that we got from our next-generation sequencing assay from Classic. And we were really happy to see that Classic performed well and could really accurately capture the fold change responses of these circuits at random. So pleased with that, we went ahead and analyzed the rest of our data, and we found that we were able to get measurements for 121,000 circuits out of 166K, which is nice, but it's still 70% of the space that we wanted to cover, which is when we turned to AI models to see if we could train an AI model with this data and predict the values of the other 45,000 odd circuits that we couldn't see from our assay. And we used random forest regression to be able to do this. It was a really nice class of models. Happy to talk about it more if needed. And to cut a long story short, it didn't work very well right at the start, but we realized that if instead of training this model with all of your data, if you train it with a very small subset of really highly accurate measurements from within your assay, so only 10% of the total measurements that we got, you can boost up your R-squared and get a model that predicts the basal and induced expression really, really well with R-squareds above or around 0.85, which is really good. So what this let us do is measure the 44.5K circuits that we weren't able to see initially, and also refit the 121K circuits that we were able to measure for a complete design space of 166,000 variants. By the way, I should mention these are contour plots, and the x-axis is the basal expression, and the y-axis is the induced expression. So really, the region that we care about is this top left corner of this space, which represents things that are low basal and high induced, so they have a high fold change response. But after this model was developed, we, as a sanity check, had it predict the same 40 variants that we had used earlier to measure the validity of our NGS assay, and found that it still predicted their behavior really, really well, which was great for us. Because then we used this completed design space of all the 166K variants to really stress test this model and built out a bunch of variants across different fold changes from these predictions, and especially about 80 circuits that were in the high fold change range. And we're really happy to see that it performed really well, and particularly allowed us to get circuits that had about 60 to 70 fold change in expression, which is pretty high for this class of circuit and wouldn't be possible otherwise. Finally, we took all of the high fold change circuits that we had from this data, which if you go back, there were only about 5,000 circuits out of 166K that produced high fold changes. So we took all of those circuits and did k-means clustering on it using the parts that the circuit had and their frequency as an input, and projected it on this 2-dimensional space, and found that there were these 3 clusters that corresponded to 3 different paths that circuits can take to achieve high fold change. And it was really interesting. These parts are kind of counter to each other and also a little bit non-intuitive. So family A, for instance, which is this green blob there, and the top half on the right, on the figure on the right here, you can see that circuits in this family had activation domains, or basically a really, really strong strength of activation, but a very low affinity for the transcription factor to actually bind the DNA sequence itself, which is kind of non-intuitive. Whereas family B and C preferred to keep both of the things kind of in the middle. So they used medium to high affinity zinc fingers and really medium strength activation domains to achieve high fold changes. So in conclusion, these random forest models allowed us to get these non-intuitive design rules out that really kind of give us an idea of how to build these circuits. And so to summarize what I've talked about here, we built a library of inducible genetic programs that turn on GFP. We used the parts as an input and used a random forest model, even though this is a neural network that I've drawn here. But we used it to predict the fold expression. And if you work in the healthcare space, you may not care about this because it's stuff that produces GFP. And you're right, which is why we're interested in taking this platform and sort of diversifying it and seeing if we can use the same principles to create therapeutically relevant genetic programs, such as things that turn on in a tissue-specific manner. And we're interested in seeing if tissue-specific transcription factors plus other microRNAs that are also tissue-specific can lead to specificity, so that when you deliver a gene therapy product, for instance, if you want to deliver it in the heart, it only turns on in the heart cells and not in the liver cells, which is where most of the gene therapies go to clear themselves out of the system. We're also interested in seeing if we can develop these complex cell therapy programs that can survive solid tumor microenvironments by expressing a CAR and then sensing these signals in the tumor microenvironment that generally lead to T cell exhaustion, and then rewiring those signals to produce outputs that help T cells proliferate better and evade exhaustion. And we're really excited to see if we can use the cell state and other modalities other than fluorescence as an input to then have an AI model predict how T cells are going to proliferate and their in vivo efficacy. So in summary, I think we have a platform that lets us do high throughput measurements, and we're really excited to work on cell therapy specific applications. And yeah, with that, I'd like to thank everyone who was involved here. So Caleb is my PI, and Ronan, who was a co-author on this paper. And I want to thank the rest of my lab, our fantastic collaborators, and some of the undergrads who worked with me. And yes, all of my cats. I should say I don't have all of these cats at once. I'm not there yet, but I foster cats in my free time. And so these are all the cats I've had over 3 years. So I'm happy to take any questions about my work or my cats. Thank you.
Thank you, Shedid.
Probably use that one. Sorry.
All right, what questions do we have?
That's fair.
Hello? Any questions? Any questions?
Maybe which is my favorite cat. That one's a good one. This one here. Yeah. His name's Sergeant Dirty Paws. Very cute, very good. Very good.
Thank you. Really great presentation. I have a question. You said that you selected subsets of gene circuits randomly to test. Do you think if you employ some kind of stratified sampling to achieve more targeted, you may have potentially better outcomes?
Yes, actually, that's a good question. So yeah, how were our circuits picked? And I guess is a little ways back. But yeah, we decided to pick these at random. And so the question is, if we decided to use a more targeted approach, would we find something that actually worked better? And the short answer is that we tried. a little bit. And in fact, when we were building or stress testing predictions from the model, some of the points, so all of these were designed by us rationally to have high fold changes. And most of them that we designed in this first round were right here at the bottom. And the reason is that we, at the time, before we had access to the data, thought that to make the best highest fold change circuit, you need the best of every part, so the strongest activation domain, the highest expression level. And so using rational design approaches like that, we got some that were like 10-fold or so on, but a lot of them just simply didn't work. And so, yeah, the AI model actually helped build all of those circuits that are in that corner. But yeah, rationally things kind of work, but not as well, I think. Great question.
All right. Thank you so much,
Great,
Kshitij. We
thank you.
appreciate it.
Thank you.